Okay, so this is going to our last talk in the morning session. We are having McPartlon from Vant AI. He gets his PhD from University of Chicago, and he's a senior research scientist at Vant AI working on protein, and protein small interactions. And he's going to talk about LATENTDOCK. I think it's NIP's paper, probably, or workshop. And yeah, let's move to the drug discovery part of the graph learning. Thanks. The microphone working? No. No? How about now? Okay. Here anyway? All right, hi guys. I'm Matt. I'm at Vant AI. I work, basically what Ali just said. I do protein small molecule interaction prediction. This is a bit of a detour from the more standard graph neural network graph learning talks. So today we're going to talk about protein docking and a method called LATENTDOCK. So this is an approach developed in collaboration with Salim Marquette, Tomas Gavner, Daniel Covenham, Alex Sergio, Zach Carpenter, Luke Ganay, Michael Bronstein, and my PhD advisor, Jimbo Zhu. So before we get into this, I'm going to first define the problem of protein docking. So this protein docking problem, the task is to predict the structure of a bound protein complex. So like the way that these two proteins might interact in your body or in a cell, given the structures of the individual components. So you get to see each individual component protein, then you want to predict how they'll actually bind in 3D and interact. And kind of this problem, at least for now, is split between kind of two approaches. There's flexible and rigid docking. In flexible docking, you want to model conformational changes in these individual components. So you might see components, how they would look kind of individually apart in your cell, and then you want to model them in their bound state. So you want to model these changes that might occur only upon binding. So for example, like when an antibody binds to an antigen, like your body simulates some kind of immune response, the actual loops on the antibody are going to change to like better match the 3D shape of the antigen when this binding occurs. So this is in contrast to rigid docking, where you're really just predicting the rotation and translation of each of the individual components. So you're just placing those as well as possible without changing the structure. Great. So why is this an important problem? So protein interactions are kind of like, they essentially control all major biological processes. So they're at the core of like antibody-antigen interactions, foundational cellular processes, like neuro processes. They also, this protein interaction prediction forms the basis for most established like drug programs and therapeutics-based discovery programs. So you want to predict maybe ways of inhibiting or ways of inducing certain protein interactions. Great. So now we'll talk about All right. Cool. So now I'll talk about some motivation for this approach. So hopefully some of you guys, at least everyone in the bio world, is familiar with AlphaFold 2. This is like the DeepMind protein structure prediction program, which is set to solve protein folding. And AlphaFold 2 is this really interesting architecture. Like you could probably get through a whole PhD just knowing this. It's really an incredible process. So it doesn't just work; it works really well. There's some really interesting takeaways that you can get just from looking at this. So first, you notice that like kind of the whole model is meant to predict a protein structure given some features. And what are those features? They're all derived from a protein sequence. A protein sequence is just some tensor of tokens. Each token has a value between 1 and 20. So that's a way to think about a protein sequence. And this protein sequence gets converted to a multiple sequence alignment, templates, all of these other things. But the important takeaway here is that all of these input features in AlphaFold2 are invariant to rotations, translations. They don't say anything explicit about 3D geometry. So this gets passed into the encoder of AlphaFold2. These are analogies that I'm making. This is not, they don't call this an encoder in the AlphaFold2 paper, so just go along with this. So these invariant features get passed to this invariant encoder. This invariant encoder is like this kind of large transformer network with some secondary updates for the pair features. I should say, like, the input is kind of your standard node features plus some edge features. These edge features are some kind of implicit representations of geometry. Great, so invariant features get passed to an invariant encoder. That encoder has some invariant latent representation of the nodes and edges. And that finally gets passed through this equivariant decoder. And that decoder produces 3D geometry. What's interesting is that along the way, like, so from the beginning to the end, there was no step where we had like these equivariant features. There's no like explicit notion of 3D coordinates. All of this is constructed by the network. So even though the input is fully invariant, we're able to predict this 3D geometry, which we'd usually think of as like only being possible with equivariance somewhere. And indeed, there is some equivariance in the decoder. However, the initialization of the decoder plus its features are fully invariant. So a question you might ask is like, why does this work? So shouldn't we need some notion of, explicit notion of geometry in order to actually produce explicit geometry? And maybe we can think about, so sorry, this goes more with this slide. So we can think about like, why does this work? Why are we able to go from invariant representations of geometry to actual explicit 3D geometry? Great. And maybe the other thing is, if AlphaFold works with implicit representations of geometry, it should definitely still work with more explicit representations. So part of this motivation is to think about the multidimensional scaling problem. And in this problem, you want to go from some similarity matrix to a set of points which kind of recapitulate the similarities. In the sense for like, if the similarity matrices are the Euclidean distances between a set of points, then multidimensional scaling can be used to exactly recover a set of points that capture those distances. So there's a very simple formula to compute the distance matrix of a set of points X. So if you have a list of points in RD, then you can write the Euclidean distance matrix of those points as a function of the Gram matrix, plus some column vectors of ones. And then you can, after writing the Gram matrix, the distance matrix as a function of the Gram matrix, you can just invert this and follow the classical multidimensional scaling algorithm, which just computes an Eigen decomposition, essentially of this Gram matrix, to recover the original X set of points. So that's one way to go from distance matrix to points. And along that way, we know kind of along the way that we haven't used any like explicit, or we've used explicit information of geometry, but we didn't have to worry about equivariance, rotations, translations, anything crazy. Kind of part of the reason for that is that this Euclidean distance matrix operator is invariant to rotations, reflections, and translations. So for any orthonormal matrix Q and like D by D matrix or translation B, the Euclidean distance matrix is invariant to Q and B. The other nice property is that this Euclidean distance matrix is low rank. So maybe you can see that in the previous slide here. So it's a function of this Gram matrix. This Gram matrix has rank D. It's the product of two rank D tensors. And this secondary term with these 1-1 transpose, which adds rank at most 2. So it's nice that we have a very low rank representation, which is also invariant to rotations and reflections. And as a consequence, so one consequence of this invariance, rotations, and reflections is that we won't be able to recover the absolute point set. We lose this kind of rigid information, but we can recover a point set that is equivalent up to these rigid transformations. Great. So kind of to summarize this, multidimensional scaling shows us the existence of an invariant mapping between Euclidean distance matrices and Euclidean coordinates, which realize those distances. And AlphaFold 2 has already shown us that implicit representations of geometry. So there exists a neural network architecture that can take implicit representations of geometry and use those to produce explicit representations of 3D coordinates. So this kind of motivates us to maybe learn in this implicit encoding space. So kind of the latent representation of some geometry encoder and use some decoder latent type decoding method to actually realize that geometry. So, right. So what does that actually give us? We're actually no longer constrained by these equivariance considerations that are typical in common kind of protein geometry related learning tasks. We're able to leverage very deep standard transformer architectures with standard nonlinearities and so on. We don't have to care about, whether or not any of our layers or any of our nonlinearities break equivariance. We're also able to leverage highly engineered like image diffusion frameworks, which have seen like a ton of engineering and they're practically incredibly useful. So yeah, a lot of progress has been made here. And rather than redefining our diffusion framework over some rotation translation space, we can just use the image diffusion framework directly because we're learning in an invariant representation. So I call this like geometric diffusion without the agonizing pain. So the other thing is we can actually naturally encode and decode other attributes apart from geometry in this invariant representation because there's nothing, there's really nothing stopping us from doing that. For proteins, we can encode the sequence, functional attributes, confidence estimates for every residue, so on. We're also able to handle much more natural representations of geometry. So it's very rare in the comp bio world that you have this exact explicit structure that you want to just like, map around. Usually you just have some kind of idea of what the structure should look like. It might be noisy, it might be incomplete, a certain section might be missing, but you still want to be able to model it. So with this type of approach, you can capture that in this distance matrix pairwise feature. You don't have to provide explicit distances. You can provide distances with some coarseness. You can also naturally mask out distances by just leaving that row, replacing this distance with a mask token or something like that. It's also very trivial to model conformational flexibility, which is something that most protein-protein docking methods can't handle, especially in this, especially in especially like with equivariance concerns. So if you're trying to predict a single rigid rotation of translation, it's very hard to actually move the underlying like atoms in each chain. So one other problem this addresses, so like why use diffusion here? Is it just because diffusion is popular and that's what gets into NeurIPS? Sometimes, but there's another issue. Yes, but no. So apart from this, like, so there is an issue with regression models for this problem, and this is kind of like we saw this a lot like way back in the early protein folding days. What we try to do is like predict a distance matrix and then try to realize geometry with like some gradient descent algorithm over these predicted distance potentials. It's not what's important here, but kind of what you see from this type of analysis is that actually like regression models like AlphaFold 2, they tend to like, the output is actually like the superposition of two or more predictions. So it's not often that AlphaFold 2 knows exactly where to place a protein. Typically it's kind of like it's split between two sites, and depending on the confidence in each site, it'll kind of move the prediction towards one site or the other. So in the final output, it has to make a single prediction. But if you look at like the intermediate state and what it thinks about like the protein structure via its like predicted histogram, predicted distance logits, it's usually kind of split between two or more sites. So you'll see like high likelihood of, for some pair of residues, between like, say here, the green and pink chain, you'll see high likelihood that, this upper site is the actual binding site, and you'll see a high likelihood that this lower site is the actual binding site. It's okay to actually like make both of these predictions when you're predicting just your histogram, but when you predict your final output, you have to decide on a site. And typically what happens is the protein gets placed between the two sites. So that's one thing, like kind of one way in which regression models fail here. So diffusion models offer a nice alternative because you'll actually be able to sample from both sites. And then you can use some like secondary ranking algorithm or something like this to decide on which conformation you want to select. Great. Yeah, so now I'll talk about the method. So this method is LATENTDOCK, and it uses latent diffusion to learn this protein-protein docking problem. So we kind of start, I'll get into the components here in a second. Great. So here, our inputs to this algorithm are kind of invariant representations of 3D geometry along with other attributes of the protein. So we derive some explicit representations of geometry from the protein's 3D structure. So this is stuff like C alpha distances. These are types of atoms at the protein residue level, and some orientations. So I said before that multidimensional scaling is invariant to reflections. One way to break this is to provide dihedral angles. So if you know dihedral angles between points, like torsion angles, then you can decide on the proper reflection. Apart from this, we have some standard like sequence information, which tells you kind of the relative positioning of the proteins. So proteins fold from a sequence of amino acids. There is a start and end, like natural language. So we have ways of kind of telling the network where that start and end is. And finally, we also provide the amino acid type. Great. So at a high level, our VAE architecture is just some encoder and decoder. And we train this before we actually train our diffusion model. Our diffusion model just recovers these latent representations, and those latent representations are converted to actual 3D geometry by the decoder. So our encoder consists of these pair-biased self-attention blocks. So it's almost like regular self-attention, but with an additional term. We'll get into that in a second. The latent representation has like kind of the standard VAE tail divergence loss. We also do some layer normalization there to kind of make it play better in standard diffusion frameworks. Finally, we keep the sequence dimension on our latent representation, which is not standard in VAE architectures. And technically, we don't have an autoencoder either because our input and output are different, but our output is just some realization of the input. So it's almost like a translation task. So yes, our encoder architecture, like I was saying, uses this pair-biased self-attention. So if you look, the standard attention formula is given here. And all we're doing is just adding some additional term. That additional term is a linear projection of the corresponding edge feature. So when you compute the attention weight for feature I, node I and node J, you also have some edge associated with it. You use a linear projection of that edge feature to bias the attention weight. So essentially what this does is it gives us a way of transferring these pairwise like geometric representations into the node representation. Okay. The decoder, we actually borrow from AlphaFold. We use invariant points attention. So kind of, this is also quite similar to the pair bias self-attention from the previous slide, but we have this additional term on the right here, which captures some like notion of geometry. So these TIs in the rightmost term are rigid frames. So they consist of some rotation and translation, and these get applied on kind of a per node level. So let's see. Right, so start with our pair bias self-attention, and then we add on this other term. So apart from like the standard like dot product attention, we also have these like point queries and point keys. And these are just linear projection of the like hidden node feature reshaped to have a trailing dimension of 3. So you can think of these as kind of like local points, that invariant local points that are associated with each node. And then we use this equivariant rigid translation to transform these points into the like frame of every node. And initially, these frames are initialized at an identity rotation and node translation. So kind of at the start of everything, or at the first layer, the points are all kind of lying at the origin. Right. So you kind of look over here. So in the local frame, you might have these points that are centered at the origin. Once you apply these rotations and translations, you get the points are transformed, and you have like the negative distance of this added to your attention weight. So the closer the points, the more they affect the attention weight. So the output of this encoder decoder architecture is a full 3D protein structure. So we model all atom coordinates, and we train this with phase loss, similar to AlphaFold 2. We also use a linear projection to recover the sequence, and we train that with cross entropy loss. Along with that, we have some per residue level confidence scores. This is like the LDDT score, which measures how well a residue's microenvironment kind of matches with the ground truth structure. So now I'll talk about the diffusion model. First, does anyone have any questions so far? Now I'm going a little fast here. Yep.
Yeah, I was just curious, did you try training the score network before convergence and it just like wasn't stable, or is this kind of person related to the network?
Yeah, so I just followed, or like the idea was to kind of follow the latent diffusion for images framework as closely as possible and kind of do what works there. Yeah, that was part of the reason. One of my favorite papers, and I like that idea a lot. And also just reading the literature for like equivariant diffusion, like a lot of these diffusion, like the diffusion frameworks they're very complex. They tend to be like unstable, very approximated. There's a lot of bugs. There's not a lot of open source implementations. They're just very under engineered. It's mostly like grad students working as a one-off. And for like things like stable diffusion or like image diffusion, there's a million implementations. You can just grab one off the shelf and apply that to a problem. So kind of wanted to make this more of like an image diffusion like problem rather than like this equivariant geometry kind of problem. Right. Yeah, so part of the reason for applying this KL divergence loss and for layer normalizing this latent representation, so kind of talked about here, part of the reason we do this is to actually like make the representation look more image-like. So this is a bit of a hack to make this work well with standard image diffusion frameworks. Cool. Okay, so yeah, now we'll talk about the diffusion model a bit. So we have a conditional diffusion model, and what we actually condition the diffusion model on. So our VAE learned to represent full protein structures, and they're given kind of full geometric information. Our diffusion model, on the other hand, only receives the structure of each like individual component. So you have some protein complex, you break this into components, and you supply the conditional diffusion model with the structure of each component, but you don't tell it how the components should be arranged. So one way of doing that is kind of aligning all of the structural templates in a pairwise matrix and masking all of the off diagonal features. Apart from this, we can also like easily incorporate things like residue contacts or ideas of like where the chain should bind by saying like basically adding these one-hot encoding features to our pair representation or our node representation to say that the proteins, the individual components should be close in proximity between these 2 points or at this certain point on the surface. right? And another way to think about this is like the score network is kind of like learning a type of matrix completion, and then the decoder is doing the multidimensional scaling. And then one other thing you can do for these template features, they don't have to be exact, they don't have to be true Euclidean distance matrices. They can be some noisy version of a distance matrix. They can be coarse one-line encodings of very like wide intervals. And you can also optionally mask the distances to design parts of the sequence. Great. So now we'll briefly talk about results. So when I talk about results here, there are different ways of evaluating protein complexes. So one is kind of all these basically have to do with this RMSD, root mean squared deviation. So you're computing some optimal alignment of two objects, the alignment that minimizes the L2 distance between the coordinates. One way to do this is over the entire complex for a protein. Another way to do this is just by aligning one of the components and then kind of measuring how far off if you align on one component. So highlighted here in orange and gray. If you just align on this, how far off are the kind of red and blue components? And then finally, you can align just on the kind of surface where the two components interact. And that gives you their type of RMSD. So for all of these, you're just deciding what you want to align. Either the entire structure, one of the chains, or just the portion you're interacting. And then DocQ score, which is what I'll be reporting, is kind of a combination of those three RMSD types plus some term which says how well did you actually recover the biological interactions that occur between these two proteins. And typically it would say like a DocQ score of greater than 0.23 is acceptable. All you have to know is that you're mostly right if the score is above 0.23. Okay. So our data set for this task, it's currently an in-house data set, but it's based on protein dimers split using a combination of what's called fold-seq and MM-seqs. So what that is a way of saying that, it gives us a way of saying that the proteins in our training set and test set kind of look, their interactions look different, and the actual sequence tokens which make up these interactions are different as well. So it's a way of kind of reducing the amount of bias between your train and test set by doing this kind of structured and sequence-based split. On top of this, so we retrained 3 kind of state-of-the-art protein-protein docking methods on the same data set. These are DiffDock, EquiDock, and DockGPT in order to do a fair comparison. Okay. So here are results for 150 naturally occurring protein dimers. So we can kind of see our method does quite well in terms of recovering reasonable, so high DocQ protein complexes. So roughly 70% are predicted with acceptable or better scores. This is in contrast to other methods. So DiffDock PP is another diffusion-based protein docking method. It does fairly well, but we're able to outperform this using this latent diffusion approach. One thing to notice here is the percentage of very high quality predictions, like very low RMSD, high quality predictions, is not as good as DockGPT, another docking method. And that's kind of because our BAE, our encoder decoder, had relatively high reconstruction error, so around 1.5 angstroms. Yes.
So in the baselines, you then consider the equivalent, which is using these graph geometry learning for the, let's say, these problems. Is it not useful in this setting, or there is another? And so in general, let's ask this more general questions. So you're using the autoencoder decoder for the learning the geometries, but why don't we use the geometry deep learning instead of the encoder decoder architecture?
Yeah, I think like so geometric deep learning can't readily be applied to this problem because there are it can be applied to a single structure, But as soon as you have two structures, well, how you decide to position those two structures relative to one another breaks equivariance. So if you want to just operate on a fixed static structure, that's fine. Geometric deep learning will let you do that. If you actually want to like fix a structure and now move some structure relative to it, that will break, like a standard equivariant architecture can't really do that, because you no longer have equivariance. Like if you're randomly choosing, say, like a starting pose, then you're just back to like standard augmented data augmentations. There are ways to get around this with geometric deep learning. So people have developed diffusion models and methods to overcome this. One is this diffdock method. So kind of the idea here is to predict this global rotation translation, which maps these final ending position. Yeah, but there are a lot of there are reasons why this sometimes doesn't work well in practice. So there's like this huge truncated like IGSO3 approximation in the actual diffusion formula, which is kind of unstable at very small time steps, very small noise scales. EcoDock tries to do something similar, predicting some key points on the protein pockets. So you can kind of think of this docking problem as predicting kind of points on each protein that will interact, and then you can think of docking as aligning these points. So that was the goal of EcoDock. But what actually tends to happen in practice is it doesn't know where the points are. It's kind of a hard problem. And maybe it doesn't have to predict like a fixed set of points. It can kind of predict points all around the protein with some level of confidence. And then when you actually do this alignment, the two proteins just kind of overlap one another. It's kind of like a low variance way of making sure that the points are relatively well aligned. So it's not actually like a biologically meaningful result, but it looks okay, say, in terms of like ligand RMSD or complex RMSD. Right. Cool. So here we just compare in terms of sampling efficiency. So I kind of like one way of thinking about this. So for this diffdock PP problem, you actually start, like in their framing, you actually start with these two structures in a random rotation translation. And the goal is to kind of move one structure, move one of the structures over to its like full docked and ground truth position. In doing so, kind of the starting point, maybe like your initial starting point, your initial random rotation translation might be important. If you're very far away from the site, it might not be able to get it there in time. So we actually show that in terms of sampling efficiency, our method tends to perform much better. So given the same number of samples, so if we only look at 5 generations for our method versus diffdock, we're able to get like significantly higher success rates. So diffdock, in practice, you actually have to sample from this a lot until you see a correct prediction. I'll quickly talk about some extensions. So we also extended our approach to other conditional protein generation tasks. So there's protein folding and protein inverse folding. So we use the same VAE because we can still recover the sequence and structure. But we kind of address each problem by doing some feature dropout in the denoising model. So for inverse folding, the task here is given a protein structure, predicted sequence. For protein folding, it's given a protein sequence, predicted structure. So I'll go quickly here. We drop out the sequence information and we use the same exact model. We just condition on these structural 3D templates, and we're able to get competitive performance with state-of-the-art inverse folding models. And finally, we apply this to protein folding, where we drop out the templates and leave this sequence information. We're able to significantly outperform existing kind of state-of-the-art folding-based diffusion models, and we're competitive with state-of-the-art monomer folding models, ESM fold developed by Meta. Yeah, thank you everyone for listening. Happy to take any questions.
Hi, that was, I have tried to talk, but with like a different, one problem that I have frequently encountered is that although I have to get all these scores, diffusion and like in this mining job on the mining side, it ends up producing a lot of flashes in the finite structure, even when they're rapidly well. But it was for the latest job that we're using for interactions. Do you suffer the same problem? And okay.
Yeah, so we compare the number of flashes with diffdock, ecodock, and our method in the paper. On average, we have like 0.1 flashes per structure. Ecodock has, I think, like several thousand. And then diffdock, yeah, I forget the actual number, but it's like 10 or so. So the nice thing here is like, so with this approach, I can find it at some point. Yeah, but so when you learn this like encoder decoder architecture, you can just apply like a very high flash loss on the decoder. So like you can train the decoder to not produce any flashes. You can make that like heavily penalize this. So anytime it sees an encoding, it knows like via adding this noise, whatever, it knows that protein structures do not have flashes. So you're enforcing this like in a model that's really easy to train. With something like some method like diffdock. So you're starting with some random rotation translation of like one of the protein chains. In order to get back to like the correct transformation, you might have to pass through the structure. So you can't like apply a flash loss in this model because actually at intermediate steps you need flashes in order to like fulfill the diffusion objective, like finding this rotation translation which moves you from point A to point B on a direct path. That direct path might contain a lot of flashes along the way, so you can't actually enforce that loss in that.
So if that end up introducing bias if you in your models, if you end up putting the hard class code, you can really do this.
If there are a lot of flashes in the train set.
Yeah, and you're avoiding them.
So yeah, if we have an example, so we use a pretty relaxed definition of flash. So we say two C alpha atoms that are within distance of like 2.5 angstroms. That's a flash. We don't actually apply this over side chains. You could, but just the C alpha atom flash tends to do pretty well. So typically you don't see it's almost impossible for two C alpha atoms to be that close.
Thank you.
Yep.
You do also.
Yeah, I checked the workshop paper that's scaling. You guys have a timeline?
Hopefully within the next couple of weeks. We'd like to release it at the same time as this data set that we use.
Any other questions?
Might be some in the chat.
Okay. Thanks. Nadir.